Okay, so now part 2 is from our learning objectives are to describe the steps for making a transgenic mouse and what techniques or substances are involved in those steps and understand the purpose of each step. We want to explain what a restriction enzyme, DNA ligase, plasmid is and how it works. Describe the steps for making recombinant DNA and what techniques or substances are involved in the steps, understanding the purpose of each step. And we need to know the applications of transgenic organisms. So, just a reminder, we are going back and we found this gene in a family because they had a bunch of members with speech disorders. That gave the scientists the comparison, and they found the sequence that was mutated in them. And so then they kept working with the FoxP2 gene, and they found that on this type of graph, it's showing, it's kind of like a family tree, but the closer two creatures are together on here are the more connected their gene sequences are. So humans are more related to chimps than they are to gorillas versus orangutan or rhesus monkeys, and mouse is way over here. But the mouse FoxP2 protein is only 3 amino acids different from human beings. So it's really close. There's only a couple of amino acids that have changed. So based on the available information about FoxP2 protein, which of the following statements makes the most sense? If it's FoxP2 gene, and remember if it's got the lowercase ox, it's the mouse. If the FoxP2 gene is removed from the mouse might talk. Feeding mice the 3 amino acids that differ between humans and mouse might enable the mouse to talk. Replacing the mutated human FoxP2 gene with a mouse FoxP2 gene is a way to cure speech disorders. Or D, putting the human version of the FoxP2 gene into a mouse might enable it to talk. So, in case you didn't know, mice normally don't talk. They make squeaks and stuff, but trying to put a mouse version of the gene into a human isn't going to really cure anything because they already don't talk. So giving them something from an animal that doesn't make any sense there. If you were going to do it, you would want to do it at least for an animal that can talk. We cannot assume that removing the gene is going to let the mouse talk because it's what enables humans to talk. Taking it away doesn't make any sense. We can't just feed them the 3 amino acids that differ, because remember that amino acids don't just get randomly put in. The amino acid that gets put into each protein has to be the one that's encoded by the RNA. So it's not that they are making these differences on the amino acid or amino acids on the polypeptide chain because they don't have enough cysteine, they're going to put in a methionine. No, they go with whatever's coded for. All of your foods contain all of your amino acids, generally speaking. There's a couple that we sometimes get from extra vitamins and stuff. But the point of the primary research article that this is based on was that they put a human version of the FoxP2 gene into a mouse, and it changed the speech patterns of the mouse. He didn't turn into Mickey Mouse, but we'll see what happens. It's more of a Ratatouille situation. So when this is just an example of how eggs develop. You get the release of the egg. It normally meets up with the sperm. Where's my mouse? There we go. Meets up with the sperm. You have a separate egg nucleus and sperm nucleus that then they fuse together. You go through a single cell, 2 cell, 4 cell, 8 cell. Then it just keeps splitting apart until it forms the blastocyst and then implants on day 6. So the vocabulary words you need to see here are zygote and blastocyst. So what they do, and there's actually a microinjection video if you wanted to watch it, is you can inject a transgene solution. So they've changed it. A solution from outside, like your human DNA stuff, you could inject it in before the nucleus has even fused. Here you have your egg nucleus, sperm nucleus. You could inject it into one part, and then they could fuse together to make a happy single-celled zygote to get implanted into a pseudo-pregnant female. You want her to be pseudo-pregnant, meaning that you've given her hormones that have triggered her to think she's pregnant, and her uterus will make the right kind of lining and stuff so that when the egg is implanted, it will start developing. You can't just transfer it at the wrong time of the month or the egg will just get passed out, basically like a normal period would. So we're going to go through and talk about what exactly happens at these steps. So we're going to draw a flow chart using some of the items in 1 through 7 to illustrate how we can make a humanized FoxP2 transgenic mouse and label where the FoxP2 is going to be at the different developmental stages, from the zygote stage to the baby mouse stage. So our options, we know that the pseudo-pregnant female mouse is going to be going on at the end. You can see from our picture here. We have the choice of working with a mouse blastocyte versus a mouse zygote cell. We have our human FoxP2 DNA, human FoxP2 RNA, human FoxP2 protein, and the mouse FoxP2 DNA. So on there, we want to inject something into the mouse cell. Do we want to inject the DNA, RNA, or protein? And do we want to inject it into a blastocyte or a zygote cell? And what happens to the mouse DNA? So give you a minute to think this through before moving forward. Yeah. Because the next question that comes up is saying, below are some alternative methods for making the humanized FoxP2 mouse that has the transgene in every single cell. So which one is going to work best? All of them say inject the human FoxP2 protein, RNA, DNA, RNA into, do we want to put it protein into both cells at the 2-cell stage? Do we want to put RNA into the zygote? Do we want to put DNA into a fertilized egg? Or do we want to put RNA into the blastocyst cavity? So, because it specifies that we want this transgene to go into every single cell, we need to hit it back before the cells have started to divide. Because trying to get it injected into one cell is hard enough. If you try to inject it into multiple cells, you're not going to get them all. It is not going to work. So you want to do it early on. And now if you give it DNA, it's going to pass the DNA every time mitosis happens. Every time the cell splits, it's going to pass a copy of that DNA to the daughter cells. If you put in RNA, that RNA would be translated into protein, but it would not get passed to future cells. Remember, RNA gets degraded quickly. It's supposed to be temporary. And if you just put in protein directly, now nothing's going to get passed to the next generation of cells. So that's going to be gone really fast. So we want to inject human FoxP2 DNA into a fertilized egg. And honestly, what happens a lot of the time is that they will, we talk about this in a little bit, they will sometimes work with nucleus in other cells and then transfer the nucleus all the way over to the egg and take out the egg nucleus completely. So they can do some different things. But just letting you know, you want to hit it at a very early stage because then every cell division is going to carry your change with it. So recombinant DNA technology is just recombining DNA from different sources. So a DNA transport vehicle is plasmids that we've talked about. So this should look very familiar to you. You should have an origin of replication, an antibiotic resistance gene. They call it a polylinker here. That's just the multi-cloning site that we talked about before. It's just where you have a bunch of cut sites that restriction enzymes can be used so that you can pop in whatever other DNA you want. So step one of making a plasmid with a gene of interest in it is that you want to copy up a bunch of your DNA. So we use a technique called polymerase chain reaction, or PCR. And all it means is you put in DNA with the appropriate chemicals and polymerases, and it cycles through temperatures, and it keeps triggering on every step here that you get the strands of DNA separate, the polymerases attach and copy over the DNA again, and then they split apart. And every single time it cycles, it doubles the amount of protein in your test tube. So within a couple hours, you have billions of copies of DNA. This is, you might have actually been hearing about PCR recently in relation to COVID-19 testing, because what they do when they do the nasal swabs is they then go mix it with the chemicals and they try to copy the viral, it happens to be RNA in this type of virus, but it still works the same way, that it can be copied. And if the virus genomic information is present, you will get lots and lots of copies of it that the machine can then detect and say, yes, it's positive for the SARS-CoV-2 virus. They specify, it's kind of little on here, these little red marks are the primers, and we can specify the DNA sequence that's going to get copied. So that's how we can tell if it's, do you have COVID-19 versus did you have another type of coronavirus that causes a common cold. So we can get in there and be very selective about what we are copying. And it happens on this. You can't get it to copy unless you have a base template. So that means that you don't get very many false positives on the test for COVID-19 unless there's a screw up at the lab where some of the test tubes get mixed or the samples are contaminated. But you can get a lot of false negatives. And that happens where when they did the swab, if they didn't hold it at the right temperature, or your chemicals weren't held at the right temperature, or you were too early in the infection process or too late, the amount of viral genetic material that's there might have gotten missed. And so you think you're good, and it turns out that it was just the very start of your infection, and then 2 days later you're sick. Like, oh crap. So that is why there is a high false negative rate on this type of testing. Usually, like in our lab, we have like some religious symbols around, and the joke is that you have to pray to the PCR gods to make sure it works right. And that PCR stands for pipette, cry, repeat. So it's not a 100% exact science. When it works wonderfully, but sometimes you have to try it multiple times to get it to work. So we've made billions of copies of our FoxP2 from human cells. Now we're going to digest our DNA of interest by using restriction enzymes. This should be familiar to you. So we've made our little sticky ends here. And now we can take a plasmid and cut with that same restriction enzyme to line up the sticky ends, use DNA ligase to glue it all together to make recombinant DNA. Nifty, nifty. See, this is why I have been having you do the worksheets. It's that each week that should be building on these topics so that you can put them together to do a full project with it rather than try to throw all of this at you at one time. So now we have our plasmid. We need to make billions of copies of our plasmid. So we're going to propagate the recombinant DNA by putting it into bacteria. And because we have a gene for antibiotic resistance, we could select on our plates to get only the ones that picked up our gene of interest. So this again should be familiar to you. Thinking back to Piglo, if it didn't pick up the plasmid, the cell can't live because the ampicillin or the antibiotic, whatever antibiotic, will kill it off on the plate. Only the colonies that are growing are the ones that have our plasmid. We can now grow up a bunch of it and extract it out and purify it, which I've done in class. And we could then inject it. So some of the bacterial cells fail to grow on the medium with antibiotics. Why is it? Is it that the gene for antibiotic resistance is not cut by restriction enzymes in dead bacterial cells? Your DNA of interest only replicates a few times in dead bacterial cells? Those cells die because the DNA of interest with the antibiotic resistant gene never got in there? Or did the antibiotic gene is not in the dead bacterial cells, but the plasmid is in there? So the correct answer is C. It's typically they never picked up your plasmid, and therefore they never got your antibiotic resistance gene. You should not be cutting with restriction enzymes inside the cells. That should have occurred well before you put it in. Your DNA of interest isn't really going to be replicating inside dead cells. And you can't get the antibiotic gene separate from the plasmid. Like, they are attached. The antibiotic gene is part of the plasmid. So that just leaves C, that the DNA of interest with the antibiotic resistance gene never got in there. And there's a video if you want to watch. So below is a list of techniques and substances involved in the steps to make recombinant DNA. And I'm actually going to pause the